out the people who published the protocol for getting microsatellites out of this species of frog. When they wrote the journal article, they left out a couple of the ingredients in the PCR reaction. And after a bunch of sleuthing around and figuring out what was missing from the recipe, we arrived at the right recipe and it went fine. So in this case, people didn't publish the complete method, probably because they didn't want anybody having access to being able to do that.
Yeah, like there are some elusive cyanobacteria people that Dr. Romley communicates with, so maybe someday we'll learn what we're doing wrong.
So,
she's like,
yeah.
email the cyanobacteria people. I'm like, cool. Discreet. Oh, wait a minute. We got a full page cheat sheet and all the freshmen are freaking out. I'm like, I don't know that I've ever had such luxury.
It's really nice to have it.
Yeah.
I mean, it was really nice to have it Fizz at least.
Like all the freshmen are outside, like, oh my god, like, we get a full page for an anatomy test. Like, I don't know what.
Yeah, I mean, to be fair, like, he has in the past, those things are kind of long.
Well, yeah. It's like well, Romley's selling my test, Romley's like, unbelievable. I never finished. I think I always left at least one or two questions blank because I just couldn't make it happen.
Tomorrow, we will have lab in White Science 219 at 8 o'clock. We will talk about data from the Hulling disk equation simulations that you ran in lab. You finished up a couple of weeks ago. That's pretty much all we're going to probably have time to do, rather than starting something new. There is a due date for that lab report on the syllabus or on the schedule. I need to look at that and make sure that I give you enough lead time. I have your literature summaries that you turned in last Wednesday graded for you. I wanted to get those graded and back to you because the next literature summary is coming up relatively quickly. I have not finished going through your lab reports because that's a due date that's going to be further down the line. I might have them all finished by tomorrow, but I might not. But I'll get them back to you at least by the end of the week, so that you will have feedback on the Bean experiment before you sit down to write the write-up on Hulling's disk equations. Yes?
As far as like sources for our intro, do you have like just use the paper that we read before we started, or kind of do you have
Certainly.
other?
Okay, so certainly functional responses, this idea that the density of prey determine how many prey ultimately get captured and eaten. That's literally what the functional response is.
Are you cool with us citing our textbook?
Yeah, sure, if it's relevant. Functional response.
For the intro of our lab report. That is not.
Oh, shit.
Right here. No shame. Senior year.
Honestly, right here. Good news though. I got a miraculous 92 on the lab.
Proud of you. That's
So
sweet.
so when I use the term functional response, this is exactly what I mean. So it has a very specific definition within ecology. This is in contrast to something called the numerical response. It's the relationship between prey density and predator reproduction. If reproduction is limited by the amount of food that you can acquire, then it's easy to see why there would be variable prey density out there in nature. And if you are living in an area that has high prey density, you would have a higher reproductive rate than if you live in a place that has lower prey density. So another way of thinking about numerical response is to say, to what degree is a predator efficient at converting the prey that they consume into the production of new predators? Both of these things are important in predator-prey models. Because a predator-prey population growth model operates kind of like competition models do, in that you're running a model that deals with, in competition, if you remember back to evolution and ecology, you have two species that are competing with one another. You run equations for both species simultaneously with one another, and the species that you're competing with, things about their biology appear in the population growth models of the other species. The same thing happens with predators and prey. Whenever you're modeling predator-prey population growth models, there are aspects of the predator biology that appear in the prey population growth model, and there are aspects of the prey population that appear in the predator population growth model. One of the things that influences how quickly predators increase their abundance in a population growth model is how they respond in a functional response way to variation in prey density. And also, when they do find prey, how do they convert those prey into the production of new predators? That ultimately, those two things ultimately have a big effect on how fast that population grows. So these are both important from the standpoint of variables that go into a predator-prey population growth model. But they're also, the first one though, is especially important in terms of looking at behavior, because these three functional responses, A type 1, which is sometimes referred to as the filter feeding model, has a particular shape. Type 2 has a particular shape, and type 3 has a particular shape. These things all are all caused by different aspects of predator behavior. Type 1, which is the straight line, there really is not much behavior associated with it. This is a situation where there's no handling time. This is a situation where there's a handling time constraint. And a type 3 response is a situation where there is generally learning associated, or switching from one prey item to another as prey vary in density. So when you think about a functional responses, the different types are formally embedded in the area of behavior, whereas a numerical response is more of an ecology kind of idea. But both of these are important in figuring out what we should expect predator-prey interactions should be at a populational level. So, all that said, Holling's work was done back in the late 1950s. There's been a ton done on this stuff. Since then, most of the work on functional responses got done in the 60s and 70s, and by the early 80s to mid-80s, pretty much people had kind of exhausted everything there is to know about these things. As a result, if I recall, there's a review in the late 80s or early 90s, I'll try and dig it up, that kind of reviews what is known about functional responses as this particular way of thinking about things is kind of just gotten to the point where we don't ask a ton of questions about this anymore because we've largely learned most of the answers about this. So I'll try and dig that up. But you don't have to have a ton of citations, a ton of resources. What you need to do is in your introduction, you need to explain what a functional response is, what the different kinds of functional responses are. You need to explain what is it about the behavior of organisms that leads to these different functional responses, and then you need to use all of that to set up why the heck we sat around in lab for a couple of weeks picking up sampling discs and putting them in Ziploc bags.
Would you recommend maybe having like a graph like that in our introduction?
Sure.
Like as an example?
Yeah. once again, if it helps you make the points you're trying to make.
Right.
I could write the introduction, as with all things, with a graph. I could write the introduction without a graph.
Right.
But if the graph helps you, by all means, figure 1 will be a graph. That's fine. With an appropriately written figure caption.
Aye.
Kennedy, did you have a question? Looks like you have a question.
Yeah, I was just gonna, like, kind of make sure, so is the daily schedule on Moodle, like, correct?
Yeah, it pretty
Okay.
much is now,
Okay.
because we're pretty much on schedule. We're in the second day of predator-prey interactions.
Yeah, so.
So I have it down.
Monday, we have Brody.
Yep.
Okay, and then we don't have the paper due until after spring break?
Yeah, that seems, okay, so that seems like an adequate amount of time for you guys to have the data, get familiar with the data, why the data look the way they do, get a report written and get it back to me. So
And
that
then,
seems like.
yeah, we just
like
have our
adequate.
second exam the Wednesday before
Yep.
break.
So hopefully we'll get through a good chunk of predator-prey interactions today, finish up with that on, wait, it's not moving around. Yeah, so we have 1, 2, 3, 4 days scheduled for predator-prey interactions. We started it last time. We'll do it today, Wednesday, and Friday. Hopefully, by Friday, we will have finished predator-prey interactions, which then sets us up to be able to talk about the coral snake paper on Monday. If we finish predator-prey interactions early, we'll just move on to the next topic, which is competition. It doesn't really change anything. The Brody literature summary will still be discussed on Monday. Which is why I wanted to get your old literature summaries, even though they came in on Wednesday, and get them graded back to you. So I have those for you today. Remind me to hand them back to you. I also have your exam, Haley. I hand it back to everybody else before.
Oh.
Everything, I got it, and I keep it to you. But it's all in the same stack of black bags.
And then, I haven't, so for the, like, literature review,
Oh, yes.
um,
I need to get topics out to you.
okay,
Wait, what?
Literature, there's a literature review, a big long literature review similar to the Lima and Dill literature review. I need to assign you topics so you can get started on that.
if you want a,
No, everybody has to do it.
oh, I thought you said that,
Yeah, that was just
that was
for-
like the stipulation.
It's the research project that you have to, have, which also I meant to remind you, on Friday. So, go to the syllabus, everybody has to write a literature review. The level of Bloom's taxonomy that you have to achieve in a literature review varies by grade. So, if you're going to review a topic to get an A, you have to basically do a good job of laying out what future directions in that area of research should be. If you're just aspiring to a B or a C, you just have to review the literature adequately and analyze it, but you don't have to do anything in terms of creating IEs, saying, these are the things that are still needed. So remember in Lima and Dill, they basically said, behavioral decisions made under risk and predation, a review and prospectus. B and C people don't have to do the prospectus part, they just have to do the review part. The prospectus is, here's what we know now, here's what we still need to go and do, here's what we still don't know, and this is what we need to go and do to figure that stuff out.
And so then what was the second part?
Oh, so if you're aspiring to a B or a C, you don't have to do an individual research project, but if you are aspiring to an A, you have to. And so something that I meant to tell you last Friday and forgot, and then we got rushed at the end, was I need to know who's going to be working with whom by Friday on your research project, so you can team up with others. And by next Friday, I need to have a preliminary idea of what you want to work on for your individual lab assignment. If you look in the lab schedule at the bottom of the daily schedule, you see that towards the end of the semester, we're no longer going to be having whole class labs. That starts at some point, down at the bottom, down the very bottom. Oh, yeah, so starting on the 14th of April, from there on out, throughout the rest of the semester, if you're aspiring for a B or a C, you don't have to come to lab anymore. If you're aspiring to an A, those lab periods are set aside for you to work on your individual research projects.
So
why I want you to start formulating ideas right now, so that you have, just because those are times set aside, doesn't mean that you shouldn't be doing something beforehand. It just means that at that point, that becomes your sole focus in lab. So
Okay.
it doesn't mean that you shouldn't do anything until April 14th. It just means that at that point, you won't have the encumbrance of lab activities to get in your way of doing that. Which is why I want you to get teams together, go through the book, look at topics that we haven't talked about yet in class, which are a lot of them. Try and figure out an area of animal behavior that you want to propose doing some work in, and then you guys need to schedule time to come and talk to me, and we'll develop those ideas and get you down the path of getting that done. And there's no lab report on that at the end, but there is a presentation. So those of you doing research on the last day of class, the last day of lab, you will see, once again, in the lab schedule is lab presentations, and that presentation basically serves as your report. I used to have you write a written report as well as make an oral presentation. I found that to be a bunch of grading to tell me the same thing that I learned from listening to you talk about your projects, and so I just got rid of the written part.
I mean, I don't know what everybody's aspiring for in here, but there's 9
Everybody's aspiring for an A except one person.
So,
You know how to talk to one another.
How many people do you want in groups?
It would be useful to have 2. It might be more useful to have 3, depending on what your project is. If you want to be a lone wolf and do something on your own, you're welcome to do that. But having somebody to split the work up with. What I don't want is all 8 of you to come to my office and say, we're a team of 8, because that's
That'd be
too
awesome.
many. Awesome, awesome. Because if you have a team of 8, 3 of you are gonna do most of the work, the rest of you are gonna sit on your butts and do nothing, and that's just human nature. So, as you think about, so the other thing is, I could assign you who you're going to work with. I'm not gonna do that. You guys know one another pretty well, figure out who you wanna work with, and then get together with that person and figure out a set of rudimentary ideas on the kinds of questions that you want to ask. The material that we've covered so far in class doesn't give you a lot of animal behavior knowledge to build on to go and start asking questions, so you may want to find yourself thumbing through the table of contents of the book. When you see a topic in the book that looks intriguing to you, you want to read about that and see if that begins to spark interest in you. And so the difficult thing both in evolution and ecology and in this class is dreaming up research ideas when you don't know a ton about the field is always challenging and problematic. But one of the things that you have is you have me to talk to, and I can help you work through these things. So I want to know who's teamed up with who by Friday, and I want some preliminary ideas by the following Friday. And these are not graded assignments or anything, I'm just doing things to kind of keep us on track and not lose sight of the fact that there's this thing at the end of the semester that's coming.
And you're just gonna, and for the literature review, you're gonna assign us topics.
And I'm gonna assign you topics.
Theoretically soon.
Theoretically soon, actually what I've done in the past is I've tried to match the topics that I give people to the types of research that they're doing, so that they kind of kill 2 birds with 1 stone. If you're doing something, for example, looking at crypsis or postmatism, I might give you a topic for your literature review that is along those lines of the importance of color pattern in predator-prey interactions or something like that. So that as you're doing your literature review, you're also steeping yourself in the area that is also the area that you're doing research in. And I can't always do that, but oftentimes I can. So the sooner I know what you're interested in, the more sensible I can make a literature review assignment for you.
More information will be given on what exactly literature review is, what you want with that.
Yeah, so there, I have a rubric for it
Alright.
and guidelines for it and everything else. But basically, you've already got exposure to what a literature review is because you sat down and read fairly thoroughly a literature review, which is Lehman and Hill. That is a literature review. It's an attempt to review all the literature on a topic in its current state. And if you want to know a good example of this, there is a set of books called the Annual Reviews And there's one on ecology and systematics. There's one on genetics. There's one on microbiology. These are little books. I should bring one next time. I'll try and remember to bring one next time. There's one in my office. They're books, and in the book is a bunch of chapters. The way the annual reviews generally work is the editors at the annual review of ecology and systematics will look at ecology and systematics and say what areas of ecology and systematics have grown enough that there's a good body of knowledge to review, but still have room for growth such that a good thorough review would be appropriate at this time. And then when they figure out what those are, they basically contact a researcher who is well known in that area, and they say, hey, would you do a review on sexual selection? And they give you 2 or 3 years to work on that, and then when you're finished with it, you have this really long, nicely fleshed out review. They typically have hundreds of references in them, but at that time period, in theory, that person has read every paper on that topic that has ever been published, every book on that topic that has ever been published, has reviewed the literature, has analyzed what we know about it, synthesized that information, and then put forth what the future of that thing should be. So annual reviews are a really good place to get a sense of what a good review paper looks like. If you're trying to get up to speed on a topic, if you can find a review that has appeared in the annual review of ecology and systematics or genetics or whatever your field is, that's a really quick way to get up and running on an area of thought in a short period of time. They usually run 20 to 40 pages, and they're comprehensive in their scope. Am I asking you to do a review that has hundreds of references and is 30 to 40 pages? No, but you should be doing your best to be hitting the high points and doing, at the very least, a thorough job, if not a comprehensive job. Does that help? Luke, you asked about the guidelines. Yeah. I'll try and dig those guidelines up and post them. They're in the file somewhere. Other questions? All right, we were going through and talking about predator prey arms races, and we kind of got to the end of this thing until the bottom here. So we were talking about predator prey arms races. Arms races are different from other predator prey interactions in what fundamental way?
There's reciprocal selection.
There's reciprocal selection. What do we mean by that? Somebody other than Alexis. Claire. Kennedy.
Reciprocal selection or?
Yeah, when she says it involves reciprocal selection, whereas other predator prey interactions do not, what does she mean by that?
Yeah, so like one's, like, organism A is selecting on organism B, while organism B is also selecting on organism A.
That is exactly what reciprocal selection is. Thank you. So that's how they get set up, and that's how they sustain themselves. What kinds of things can happen that brings predator prey arms race to an end? We spent a lot of time on this at the end of class last time.
Yeah, like a trait, when a trait becomes fixed, like there's no more genetic variation.
When there's no more genetic variation, what else can happen that puts an end to a predator-prey interaction?
Extinction.
Yeah, the extinction of one or both of them. It's a weird way of thinking about a predator-prey arms race coming to an end, but that certainly ends the predator-prey arms race. The last thing, what other evolutionary processes can you think of that can lead to this type of ever-escalating selection? This is the one question we didn't get to last Friday. We've already talked about these organisms in this class once before. At least I don't think we got to this last Friday, did we? I don't think we did. I described one of these species in terms of an arms race. Do you guys recall widow birds? What's interesting about widow birds?
Their eyes.
Eyes are on the sides of
No, that would be the woodcock.
Oh, yeah.
Widow birds are the ones with the long tails.
I was like, oh.
Occur in the plains of southern Africa. They have really long tails, they have these elaborate courtship dances. What did I say was the reason for males having such long elaborate tails? Female widow birds thinks it's really, really sexy. So as you are a male widow bird out there, you have higher reproductive success if you have a longer male than your peers, because females are choosy. But if there's a genetic basis to choosiness in females, the female offspring of choosy females will be more reproductively successful because they will produce male offspring that have long tails because they chose male offspring to mate with. And so you end up getting female preference and male trait genetically tied to one another, such that females choose males with long tails. They produce not only males with long tails, but also they produce females who are choosy, which then brings about a more choosy generation of females, which then would bring back about a more long-tailed generation of males. And then the whole thing just escalates because females are selecting on males, and in the process of that selection, they also essentially are exerting selection back on themselves to a sense. Yeah?
Could it ever work that the choosiness of the female works against their reproductive success?
Yeah, so this is another one of the situations. There might come a point where male widow birds have tails that are so long that selection against male widow birds from a predation standpoint is greater than selection on the strength of selection on males from the standpoint of female choice. And at that point, those two forms of selection are acting against one another, leading to, once again, this form of constraint, such that one type of selection is acting against another type of selection. And then what you should start to see is stabilizing selection, where you have mortality selection from things like predation and sexual selection from female choice that just makes the phenotype of male widow birds more and more narrow. And so the mean isn't moving around, it's just becoming less and less variable over time. Does that make sense? So sexual selection is another place. As a matter of fact, when Fisher described this form of runaway selection, when R.A. Fisher, geneticist back in the 20th century, described runaway selection, his example was actually sexual selection. And it wasn't until later that we began to start thinking of some predator-prey interactions as having the potential to also involve this runaway selection. So predator-prey arms race is just one specific example of a runaway selection scenario. So I gave you this figure when you were writing your paper about bean selection, because this is actually written from the perspective of predator-prey arms races. Predator-prey arms races are more likely when the consequence of encountering a prey phenotype, and that has a large effect on predator fitness. So if prey phenotype in this case is tetrodotoxin level in newts, from hardly any tetrodotoxin to a lot, if that has a large effect on predator fitness, then you should expect a predator-prey arms race to get up and running. But also, the predictability of that consequence, i.e. how much variability there is around that line, also makes it more probable that you're going to get into an arms race if that predictability is very high, meaning that there's very little variance. Here, the consequence is the same, but it's more of a Russian roulette kind of thing in terms of whether or not an individual predator experiences the full force of that consequence. Here, the consequence is relatively small, although the predictability of the consequence is pretty predictable. Here, the consequence is not very large, and also there's a lot of variability. You would expect prey selection on predator to have the least amount of effect here. You would expect it to have the most amount of effect here. And if you want a further elucidation of this, go and read that paper that I put on Moodle, Brody and Brody 1990. It's a bioscience article. So they wrote this in 1990? That should be 1999, actually. Not 1990. I need to fix that. They wrote this article when they were partway through this process of exploring the predator-prey interaction between Taurika newts and Phanophis garter consequences large and predictable, consequences small and not predictable. These represent different kinds of scenarios under which you can expect varying degrees of the probability of predator-prey arms race starting up, and this is the one where you should definitely expect predator-prey arms races to start. So, in your book, there's a little, I think it's in a box. They talk about whether or not, they try to make the distinction between adaptation and storytelling. I forget where it is. Luckily, Luke has his book here. Where is it? Oh, it's at the top of page 86. So one of the complaints about, it's one of my complaints about nature documentaries, and it's also a complaint about research in behavioral ecology, is that oftentimes we see things out in nature, we build adaptive stories that presumably explain these behaviors that we see out in nature without ever having done any research to determine whether or not these behaviors actually have adaptive value. And so they make this distinction between whether or not something is truly an adaptation or whether or not this is just a convenient and plausible story that we tell ourselves. The problem with this is oftentimes it's difficult to identify whether or not something is truly an adaptation without quite a lot of research going into it. And so don't get trapped as you go out and make your own observations of building plausible stories about the behavior of the organisms that you're observing if you don't have some really good evidence to say that that's something more than just a story that you might be telling. It has always been assumed that horned lizards have long horns because that is an anti-predator mechanism. And it's plausible, it makes sense, but nobody tested it until the 1990s. You read a very short paper that appeared in Science when you were freshmen, when you were first years, that basically tested that for the first time. It's 1994 or 2004? 2004, sorry, it was 2004 before anybody actually tested it. And so, even though we always assumed that that was the function of those horns and horned lizards, nobody had actually given it a hard experimental test until 2004, when they went out and looked at the horn sizes of living horned lizards running around the landscape and looked at the sizes of horns on lizards that have been caught by shrikes and impaled on thorns. And they found that those horned lizards that had been caught and impaled on thorns by shrikes had actually shorter horns on average than horned lizards that were running around not being impaled by shrikes on thorns, indicating that at least where shrike predation is concerned, horns are useful for increasing fitness. In which case, it's now firmly an adaptation against bird predation, whereas before it was just a plausible story. You guys remember that paper? So, we're going to diverge from the book because the book talks about predator-prey arms races, and then it starts to talk about mimicry, which is not necessarily a predator-prey arms race because it's hard to imagine how mimicry exerts reciprocal selection back on predators. So let's talk about an actual predator-prey arms race. Some of this is material that we covered in evolution and ecology, so I want to run through this relatively quickly, but completely so that you kind of understand what the whole thing is about. I got introduced to this system because E.D. Brody Jr. was the department chair at the biology department at the University of Texas at Arlington when I was a PhD student there. I was in charge of a speaker series where we got to invite a bunch of really good speakers into the department, and Bush Brody III was one of the speakers that we brought in, and it was all during this time period when they were beginning to explore this system at the beginning. You guys, how many of you are in Tri Beta? Tri Beta? biology department is willing to pay for a speaker that TriBeta designates. The nice thing about asking people to come and speak as a student is they are much more likely to come and speak than when they're asked by faculty members, because faculty generally want to talk to students. And so we were able to get in a lot of really high profile biologists to come and talk at University of Texas at Arlington, because we were a graduate student speaker series, not a faculty member trying to fill out a departmental seminar series. And so we talked about this in the department meeting last week. We have put out the offer to TriBeta to choose a speaker or two that we will attempt to coax to come in. As a TriBeta speaker, the department will pay for it. You don't have to pay for it out of TriBeta coffers. But what we need from TriBeta is we need a suggestion of who you guys want to hear speak. And so think about the research that you're doing, think about people that you read, et cetera, et cetera. If they have a compelling story that you think other people would benefit from here, get together with your TriBeta cohorts and submit a name to Dr. Judd. He'll communicate it with Dr. Reynolds, and we'll see if we can get those people in. Little Butch came to talk. I got exposed to all this because these two people were associated with the University of Texas at Arlington. The first of these papers came out in 1968 when Butch Brody Jr. was a student in Oregon, and he went to his professor and said, hey, I want to do undergraduate research, which was really uncommon in the late 1960s. And this faculty member that he approached said, I don't really have anything that I'm working on right now, but there's this story that appeared in the paper some years ago about these fishermen who were found dead at their fisherman's camp, and nobody knew how they died, but the only thing that shed any light on it at all was that they found a newt that was boiled inside of their coffee pot. Presumably they had gone down in the stream, scooped up some water, put it on the fire, dumped the coffee grounds in there, drank the coffee after having boiled the newt. The coffee water was presumably filled with some toxin, and they all drank it, and they all died. There were 3 of them who died at this camp. He said, find out if the newt had killed the fisherman. And so Big Butch Brody Jr., he went and he got a bunch of newts, and he fed portions of newts to every vertebrate that he could find. And it killed almost all of them relatively quickly with relatively small amounts of newt pieces. One of the species that actually didn't die was Thamnophis hortalis, and then he graduated, he went off to graduate school, he wrote a little paper in the Journal of Herpetology, and then he didn't think about this particular system again until the late 1980s, when his son was now a student at the Department of Ecology and Evolution at the University of Chicago, one of the best evolutionary biology schools in the world at the time. They then started thinking about this because Butch Brody III went to Oregon to do some work on stripedness and reversals in snakes, found that some of the snakes that he was working on were eating newts occasionally and surviving the event, while other snakes were eating newts and not surviving the event. This got him thinking about this story that his dad told him. He approached his dad about it and said, hey, what about this thing? Tell me more. And they cooked up this collaboration that goes on to the present. So in this first paper, tetrodotoxin resistance in garter snakes and evolutionary responsive predators to dangerous prey, they went and basically looked at a population of Thamnophis hortalis in Oregon that is sympatric, meaning it co-occurs with newts. A population of Thamnophis organoides that is found at the same place, it co-occurs with newts. And a population of Thamnophis hortalis that occurs in Idaho where there are no newts. Thamnophis hortalis is a wide-ranging species. I have Thamnophis hortalis under my porch at my house here in town. They overwinter under my porch. So what's the difference between Thamnophis hortalis and Thamnophis organoides, if you guys can remember? Yeah, I'm asking you to remember stuff from a long time ago in some of your cases.
One is striped, one is that?
No, that's
one?
Thamnophis organoides. So the stripe the striped and checkered snakes are all Thamnophis organoides. So Little Butch was in Oregon working on Thamnophis organoides and occasionally running into Thamnophis tertalis as a result of just being out there catching snakes. Luke, you should remember this, you've had it recently, more recently than everybody else in the class.
It's not the size of it.
Nope. What does Thamnophis tertalis eat?
Oh, one of them eats newts, one of them doesn't eat newts, right?
This species specializes on newts. This species specializes on slugs, worms. So both of these species live in an area where newts occur, but one of them regularly eats newts and the other one doesn't regularly eat newts. Thamnophis tertalis from Idaho doesn't ever eat newts because no newts occur there. And so what they did was they injected varying levels of tetrodotoxin into the snakes and then raced them down a racetrack to see how much of their baseline speed did getting injected with tetrodotoxin cause them to lose. So if you're up here at 100% baseline speed, after being injected with tetrodotoxin, it didn't slow you down at all compared to what you would run, what you would slither down a track without tetrodotoxin. But as the concentration of tetrodotoxin goes down, the percent of baseline speed that you have after injection with tetrodotoxin goes down. But that goes down at much lower concentrations for things that either don't eat newts because they don't eat them, or don't eat newts because they could eat newts but they don't live in a place where newts occur. A species that lives in a place where newts occur and eats newts has a much higher resistance because it takes way more tetrodotoxin to slow them down after they've been injected with tetrodotoxin. So this is how they're going to go through and assay whether or not snakes are resistant to tetrodotoxin is inject them with a concentration of tetrodotoxin and then see how fast they are after they've been injected with tetrodotoxin. Based on this, would you say that there's a possibility that there's a predatory arms race? Why?
You have two groups of Sertalis, only one survived the new,
And these guys are surviving, they're just way, way slower. Would that maybe have a fitness consequence for these individuals?
But there's, like, it wouldn't be reciprocal selection there, though, would there?
Why not?
What would they be selecting against the newts for?
So if you're a Thamnophis tertalis and you encounter a newt that is not very toxic, you eat the newt and you survive. That might drive newts to become more toxic. As the newts get more toxic, they exert selection back on the snake. And then the snake has reduced locomotor ability after consuming a newt. That may have consequences for its ability to get away from a predator. As a result, they're exerting selection back on the snake. If there's variation in resistance in the snake population, only those individuals that are most resistant will survive that encounter, and then they will now be more resistant as a population that then exerts selection back on newts to be more toxic.
But in order to be affected by the newt tetrodotoxin, don't they have to eat the newt?
They have to eat the newt, and they have to eat the newt.
So that would already like decrease the newt's fitness but then how, I mean, like if that newt.
That newt wasn't toxic enough. It perished, which
Right.
means who's left in the population?
The toxic ones, but even if they're more toxic, they're still gonna get eaten, and that's how the toxicness gets to the snake. And kills it too, right? So even the toxic newts would die, cause they'd get eaten, but then the snake would too.
Would they?
That's what I'm asking.
Yeah, so it's been too long for you guys to remember the ins and outs of all of this.
The snake doesn't die though, right? It just,
The snake doesn't have
Gets
to die.
paralyzed, right?
It gets paralyzed. Oh, and I have a tetrodotoxin story to share with you also.
Didn't you bring the new to class one
Yeah.
day?
I did, I don't have any more left in the lab.
which
Hmm?
Gave them.
Well, they eventually die, I mean, that was like, it was like 4 years ago.
I think you had one in like the Moodle course picture.
Mm-hmm.
Yeah.
Yep.
Do you name your little animals that you have?
Yes, they have animals like article it's in, but I do remember the story they told, and it does appear in one of their articles. But there was one day that Little Butch was out in the field collecting snakes, and this will address your question so cleanly that you will be shocked and surprised. Out in the field collecting snakes, he ran across a garter snake that appeared to be dead. Decided that he would bring it back and try and ascertain what killed it. Put it in a pillowcase, put a knot in it, attached it to his belt, which is what you commonly do with snakes. Finished collecting all of the snakes that he needed for that day, all the ones he could find for that day. Got back to his camp and started processing through everything. One of the first things that he started to process was the dead snake, because it was the dead snake while the other snakes were alive, and if the dead snake stays dead long enough, it starts stinking up the place. And he opens up the bag, looks inside of the bag, and now, instead of just a snake in the bag, there is a dead snake and a newt that had evidently been consumed by the snake, and then after the snake succumbed to tetrodotoxin, the newt climbed out of the dead snake's mouth.
Adorable.
So, just because you get consumed by a predator doesn't mean that you are killed and eaten. In this case, the act of being consumed killed the predator and allowed you to crawl out of the dead snake's mouth. So when we go back to the predation sequence, handling, the last phase here is about killing and eating. But snakes swallow their prey whole. Oftentimes they're swallowing their prey while the prey is still alive. And so one of the ways of avoiding being killed and eaten is to get eaten, have a toxin that immobilizes your predator, and then you escape because they're on their way to death and you're still alive. So does that answer your question adequately?
You said like gills or something?
No, they don't have those.
How do they survive, how do they hold their breath?
They have relatively low metabolism, they breathe through their skin.
How do they breathe?
If you have low metabolism, you don't use a bunch of oxygen, so you don't have to be breathing very much. Yeah.
What was the utility of using, the Simpatric one that eats slugs and the one that doesn't? Like, why do they need two snakes that don't eat the newts?
Because you could have acquired resistance because of something other than something in your diet. Maybe there's something in the environment. If you go to old copper mines and look at fruit flies, I know this because my partner does this, and she drags me to copper mines, some of the most scenic copper mines in the world. We have been to flies just flying around in areas where there have historically been copper mines are much more resistant to copper than even fruit flies on berry farms and things like this, where they use insecticides where copper is one of the active ingredients in it. The fruit flies at copper mine sites are way more resistant to copper than those on berry farms, and those are more resistant than ones that are just flying around in nature, not on berry farms. And so just being in the environment can make you resistant to something if it's just ubiquitous in the environment. So is the resistance to tetrodotoxin due to the fact that you eat the potential source of the tetrodotoxin, or is it something that is just hanging around in the environment that you might come into contact with and as a result get resistance to? So because these two things come from the same fields and pastures and forests as one another, if one of them is resistant and the other one isn't, the one that is resistant is the one that also eats the newts, then it identifies the newt as actually the source of the tetrodotoxin that is supplying the selection that has led to the evolution of resistance to tetrodotoxin. Newt, worm, eats amphibians, but doesn't eat toxic amphibians because toxic amphibians don't occur there. So there are two ways of not evolving resistance. Don't live in the same area that a toxic amphibian lives. Or the other way is if you live in a place where a toxic amphibian is, you can avoid evolving resistance by just not eating that particular prey item. Thamnophis garter snakes are well known for dietary specialization. Butch's major professor, Little Butch's major professor, Steve Arnold, published these studies back in the 1980s looking at chemosensory behavior in garter snakes. If you stick a swab in front of a garter snake, like a Q-tip, if you dip that swab in saline for 10 seconds or so, tongue flick rate goes up. But as the snake realizes that there's nothing of interest there, tongue flick rate goes down really quickly. Tongue flick is how they gather information, sensory information about their environment. Scent particles, chemical particles get stuck on the tongue, they pull their tongue into their mouth, and then their nasal mucosa is exposed. So while we have a hard palate and then a soft palate behind that, that keeps our nasal cavity separated from our oral cavity, snakes have a hard palate, but then their soft palate is gone, and they have an opening between the mouth and the nasal mucosa, and they take the forked tongue, the tips of the forked tongue, they wave them around up there, and it enhances their ability to smell. And so tongue flicking in snakes is a kind of an honest indicator of how interested a snake is in something. And so you can take a swab, a long Q-tip, dip it in saline, stick it in front of a garter snake, it'll flick for a couple of times, then lose interest because there's nothing of interest there. Take a swab, rub it on a worm, stick it in front of a garter snake, and if the garter snake eats worms, the tongue flick rate will go up, and it will go up and be sustained, and in some cases they'll actually attack the swab because they think it's a worm, because their sense of smell is telling them that it's a worm. Take that same swab that you rubbed on an earthworm, put it in front of a newt-eating snake, it will tongue flick for for a limited amount of time and then lose interest in it because it's not registering as anything of interest to them. Same thing, rub a newt on a swab, stick it in front of a worm-eating snake, it will lose interest in it. Take a newt swab and stick it in front of a newt one, and it will tongue flick like mad. So Steve Arnold showed that these snakes have kind of genetically determined food preferences that are easily measurable by looking at tongue flick rates when presenting with different olfactory stimuli. So garter snakes are well known for being dietary specialists because your choice is to either evolve resistance or to go eat something else. Well, if you have an evolutionary history where you eat amphibians on a regular basis, that has then canalized your sense of smell such that you really only respond to amphibian-like scents, and then you find yourself in an area where the most abundant amphibian in the environment is a toxic newt. You can't really change your behavior because you have a long history, evolutionary history of specializing on amphibians. So then as you expanded your range westward and got into a range where these newts occurred, you didn't really have very many options because you had already gone down this phylogenetically constrained pathway of being an amphibian eater. That's a really
Yeah.
long answer to what seemed like a simple question.
No, it's okay.
Yeah.
How would they know it's a genetic trait in that situation unless they, like, if they took them from the field,
Yeah.
wouldn't that just be an adaptation as opposed to a genetic trait?
An adaptation is a genetic trait. You're thinking like of a physiological adaptation, which I use
Okay, yeah,
adaptation.
maybe that, okay.
Yeah, so it could just be that they become acclimated
Yeah,
to
to what's
that.
in their
Yeah,
environment.
sure. So one of the things that you do with this is you generally do this with family members. So when you go out into the field and collect snakes, you don't collect individual snakes. You generally collect a mother who has a bunch of babies inside of her, and you can feel them, you can palp them and see how many babies are in there. You know, if you have a gravid snake, when you have that gravid snake, you wait for it to give birth, you test the babies, and then you look and see, is there variation among families in something like resistance? Generally, that variation among families is going to be due to family-specific underlying genetic background, which basically means that it's a heritable trait. And they go into the genetics of it later.
That
But
is-
it's a good question. So, in this paper, they actually calculate broad sense heritabilities because they have the moms and they have the babies, and they have the family means, and you can calculate broad scale heritabilities by just looking at how much variation in the trait, i.e. resistance, is due to differences among families, and that's divided by differences within families. And so that's a simple variation among families divided by variation within families. So 328 neonates from 23 families, that's how they did that. The second paper that you guys read came out in 1999. They were interested in whether or not there was a cost of exploiting this dangerous prey. So some populations are resistant to newt toxins, tetrodotoxin. Other populations are not resistant to tetrodotoxin. Why does the snake not just evolve super resistant such that they are way more resistant than the most toxic newt that they might encounter? This is one way in which a predator-prey arms race can come to an end. One species evolves out of the predator-prey arms race. Well, one of the ways that you can't evolve out of the predator-prey arms race is if there's some cost to, in this case, having resistance. And so they were interested in measuring that. And the way they did this was by looking at baseline speed, regressing post-injection speed on baseline speed. The dotted lines are a one-to-one line so that there is no cost associated, and the solid line is the actual best fit through the points, looking at individuals and looking at baseline speed and post-injection speed. And back during E&E, I gave you this figure. So if you race a snake and measure their baseline speed, and then inject them with tetrodotoxin and measure their speed after their post-injection speed, if there's no effect of tetrodotoxin, this will be a one-to-one line. Somebody, slithering 5 meters per second will also slither at 5 meters per second after injection. 10 meters per second, 10 meters per second after injection, whatever these numbers do. I don't know of any snake that can go 10 meters per second, but maybe it's 10 centimeters per second. But this one-to-one line represents a situation where tetrodotoxin doesn't have an effect on the snake at all. So if there is an effect, what would you expect to happen to this line? Is the slope going to change? Is the y-intercept going to change? Is both of them going to change? If there's an effect of tetrodotoxin but no trade-off, what will happen to this line? The y-intercept will change. Snakes that were fast are still fast. Snakes that were slow are still slow. What's happened is just this line has dropped down such that the distance between this line and this line represents the effect that tetrodotoxin has, but that effect is the same across all baseline speed snakes. If there's a trade-off, what would you expect to happen to the line?
Slope would change.
The slope would change. The slope would change, and if it's a trade-off, the slope would change such that fast snakes, baseline speed, are slower after they're injected. Slow snakes, baseline speed, are less slowed down by tetrodotoxin afterwards. So if you have a cloud of data that relates the speed of snakes, baseline speed and post-injection speed, the average X value of that and the average Y value of that, this indicates if you're at 11 centimeters per second, you on average get slowed down by 6 centimeters per second when you're injected with tetrodotoxin. But then this would be a one-to-one line through that point. So this would be what you would expect if there was an effect of tetrodotoxin, but no trade-off. But you can see that this line doesn't fit the data very well. And so you can measure the trade-off by asking, is the slope of the line a slope of 1, or is the slope of the line, of the best fit line, something less than 1? And in this case, the slope of the line is less than 1, such that faster snakes are more affected by tetrodotoxin, and slow snakes are less affected by tetrodotoxin. And so this is one of the ways in which predator-prey arms races can be perpetuated, is when the evolution of one of the players is slowed down because there is a trade-off. If you are really, really resistant to tetrodotoxin, you're going to tend to be slow, such that, if you're really, really, if you're really, really slow normally, you tend to be more resistant, and if you're really, really fast, you are less resistant. That means that there are two things acting here. There's selection exerted on you by the newt that you're eating, but there's also potential consequences of getting eaten yourself by other things. So being really resistant, if that also makes you slow, that means that under any normal day when you haven't eaten a newt, you're just generally less able to escape predation than a faster snake that on a normal day without eating newts can get away from predators faster, but then as soon as you eat a newt, you're seriously debilitated as a result of that.
If the slope isn't 1-to-1 and it never changes from that deviation, does that mean that eventually the arms race is gonna end because one will out-evolve the other?
No, not necessarily. It just means that there's no trade-off. So if the process is gonna be slowed down, it's gonna be slowed down by something else. And it could just be that both of them are evolving really fast. You're in a red-green hypothesis sort of way, and you just have very fast escalation of both newt toxicity and snake resistance. This allows it to slow it down such that it plays out over a much longer time period in evolutionary time because the trade-off exists. It doesn't have to have a trade-off, it just so happens that in this case it does. So the 1-to-1 line is the effect of tetrodotoxin with no trade-off, and the solid line is the effect of tetrodotoxin with a trade-off. And what you see in all of these panels is that this line is a significantly lower slope than the 1-to-1 line, and this is when you look at it for individuals, and this is when you look at it for family means, which also indicates that this trade-off is heritable as well. Is this all coming back to you?
Somewhat.
Somewhat. Let me know if I'm going through this too fast, because I'm blazing through this. We've, in E&E, we spent a whole class period on the first paper, we've spent a whole class period on the second paper. And we've spent 15 minutes on the two of them. All right. So, the interface at which this predator-prey interaction occurs is at the level of the sodium channels in the snake. So it's the sodium channel that interacts with tetrodotoxin. And so when you look at sodium channels, sodium channel protein has 4 domains, 1, 2, 3, 4. It basically is 4 proteins all stacked next to one another. They're connected to one another by these cross-linkers. It's a transmembrane protein. Sodium channels go all the way through the membrane, allow sodium channels to flow from outside the membrane to the inside of the cell. In the process of doing this, it depolarizes the polarity of that membrane. And then there are sodium-potassium pumps that pump those sodium ions back out to repolarize that membrane. And then when a sodium channel opens up, that sodium comes back through, changes its charge on the membrane. Domain 4 is this last domain here, and this is where all of the mutations have been found that lead to extreme resistance in snakes. And so here is a phylogeny of a bunch of garter snake species. There are 3 of them here that all have really high levels of resistance. All 3 of those share a single mutation with one another. 2 of those share a single mutation. This mutation at amino acid residue 1568. This one has less resistance than the other 2, but still has really high resistance. Has a novel mutation here at amino acid 1276 in this long string of amino acids. As it turns out, this amino acid change is very interesting from a functional standpoint. In the paper that I'm referencing, they had Bear Lake, which is in Idaho. It's the original Idaho population they looked at. Willow Creek is in southern Oregon. Benton, which is in kind of north central Oregon, and I forget where Warrenton is. But this represents no resistance, weak resistance, strong resistance, and the strongest resistance as indicated by the mouse-adjusted mass units of tetrodotoxin that these populations can sustain. And what they found when they took the genetic code for sodium channels, so this is ultimately a long single protein. It's encoded by DNA. And these are the amino acid residues that are specified by the DNA that would build this protein. What they did was they basically used human DNA for most of this to build a sodium channel. And then they took that human DNA, lopped off the end of it, and replaced it with snake DNA using standard cloning techniques. And then they injected that DNA into frog eggs, let the frog eggs express this altered DNA, this chimeric DNA, and then tested those frog eggs to see what the presence of sodium, what the presence of tetrodotoxin did to their ability to open up and allow sodium ions to go through. So take human DNA, add a little bit of snake DNA of these different populations, put that in a frog egg. The frog egg builds the sodium channels and then tests the resistance of the sodium channels to varying concentrations of tetrodotoxin. We are out of time. We will pick this up on Wednesday. Questions?
Whoopsie-doo.
All right, I'll see you guys
Oh, wait.
tomorrow. Yeah. Oh, yeah, I have stuff for you. Don't I? I say I have stuff for you. Do I really?
Something.
What did I do with it? Haley,
Yeah.
Zan, and Jack. Jalen. Luke. Alexis. Logan. Claire. Kennedy. Caleb. Keep track of your grades. There's not really a way to do this on Moodle because you can't have on Moodle a bunch of numeric grades and then pass-fail grades. So this is your first literature summary. If you're shooting for an A, you needed to pass this. If you're shooting for a B or C, you didn't have to pass this, but the sooner you pass them, the better off you are. If I have written on there that you barely passed, you need to pay attention to those things that almost took you down. Also remember that if you fail because of a writing specification, you have 3 tokens that you can turn in to convert that writing specification failure into a pass. All right, see you on tomorrow.
See you on